In regenerative medicine, a robust protocol of differentiation of pluripotent stem cells toward a specific neural cell type is a valuable tool. In our case, these protocols were very useful to study both glioblastoma development and Parkinson's disease. So first, the generation of three-dimensional organoid has the advantage of closely mimicking physiological development, and also the production of size-calibrated neurospheres from pluripotent stem cells has high reproducibility. The following procedure will be demonstrated by Manon Locatelli, a PhD student from our lab.
"24 hours before starting the 3D culture, replace the HES-SO medium with serum-free medium supplemented with 10 micromolar ROCK inhibitor. The cells should be at 60% confluency. The next day, detach HES-SO colonies as single cells by removing the medium and rinsing with calcium and magnesium-free PBS. Then, add 5 milliliters of enzymatic dissolution solution and incubate at 37 degrees Celsius for 1 to 2 minutes. Afterward, collect the cells in serum-free medium with 10 micromolar ROCK inhibitor and centrifuge the cells at 300 times G for 5 minutes. After centrifugation, check the size of the cell pellet. Remove the supernatant and count the cells in 10 milliliters of serum-free medium supplemented with 10 micromolar ROCK inhibitor. In parallel, rinse the microwell plate with 2 milliliters of serum-free medium per well and centrifuge the plate at 1200 times G for 5 minutes to remove all bubbles in order to prevent neurosphere.